I've got some slides to share.
Yeah. So let me just use share content. Screen share's not automatically doing it. Oh, sorry, 1 second. Yeah, there we go. Can you guys see my screen okay?
We can. Right. Do you want to maximize the...
Yep, that's fine.
Let's make sure we are recording. Hang on. Yeah, we're recording.
Okay, excellent.
Okay, away you go.
No problem. So hi everyone, I'm Chris.
And you use your mouse key as a pointer.
Okay, perfect. It's just a very short talk on a little bit about what we do over at Ivan's group in Babs, and the main project that I've been working on. Initially, I came on as a research technician under him, and it was a diagnostic project for a parasite disease called CHAGAS disease, which I can explain a bit about now. And then we started to go more with the second grant into the therapeutic development side of things. But today, I think I'm going to focus on the diagnostics mainly, because it's wrapped up in a nice bioarchive paper that we're just basically putting out into journal at the moment. So just for the background, there we go. Just for the background side of things, the Chagas disease is the focus of the research project, and it's also known as American trypanosomiasis. I can never pronounce this, trypanosomiasis, but it's South American endemic parasite disease caused by this little guy here called a Trypanosoma cruzi parasite. It's a blood stage parasite, and it's mainly transmitted by this vector here called the tritamine bug. It's actually quite prevalent and endemic in South America. It's an interesting parasite disease because there's no vaccines. The only cures are, quite, let's say, nasty antiparasitic treatments, and they have to be administered very quickly to actually have an effect. So they're very ineffective once the infection has taken place. And what happens in this case is it basically is a condition that has a blood stage and then a chronic. So the blood stage is an acute infection, and then there's a chronic infection stage. You get this acute infection upon the parasite circulating in the blood, and that normally lasts about, let's say, 4 to 8 weeks. And that is when, funnily enough, this is asymptomatic at that stage. You get very mild symptoms like maybe flu or cold, but nothing super serious that people are quite, it's very obvious what it is. And after that, the parasite essentially goes through stage changes and burrows itself into tissues where it replicates. And that can last decades, years to decades, without any new symptoms appearing. And then what you have is very, very severe. In about 30% of cases, chronic complications relating to, the heart, the gastrointestinal system. And it basically kills, well, approximately about 10,000 deaths a year, they suspect. And we think there's about, WHO has predicted about 7 million people infected globally with Chagas. It's also spreading to other areas with migration and people moving around, because it can be blood, passed on by blood. So, transfusions, things like that, affect its transmission. It's also just been made endemic in North America as well. So that's gaining a bit of interest in treating this condition. And it's known as like the silent killer for these reasons, because it's, it doesn't necessarily show up until it's too late, and people call it a ticking time bomb. So there's an urgency, real push for diagnostic and therapeutic solutions. Diagnostic tools often require lab, molecular approaches, PCRs, ELISAs, blood work, continuous monitoring. And that is very expensive, especially because it affects disproportionately rural areas in South America that don't have access to these kind of things. So, so we are trying to look at very affordable, reliable ways of diagnosing this at blood stage very quickly. And we think the approach to take, which could be quite good, is like a lateral flow test. So that's what we were trying to work on. So the group, so before I go on to the diagnostics, we'll just talk about the group. In the group, we generally focus on 3 areas of biology relating mostly to parasite disease. In the therapy approach, where we look at things like monoclonal antibody discovery, nanobodies, trying to actually map and go down the structural route of these molecules and looking at whether they are, you know, on target and have really nice kinetics so you can use them for a therapeutic approach. Yeah, here it's like the pipeline we tend to go through from is discovery to preclinical, essentially. So you like target validation, parasite assays, and with collaborators, mice models where possible. Also, collaborators is parasite cell invasions, because this is a Cat3 parasite. So it's, you know, that has to be handled by the collaborators, unfortunately, but we're working on that. We also have a computational approach to therapeutic and diagnostic tools. In particular, Lee should talk about this, but these things called AI binders, which are, you know, all the bells and whistles at the moment. And we've actually gone through the pipeline, designed panels, tested them in parasite cell invasion assays, and shown, you know, a considerable reduction compared to some of the antibodies that we work with. So we actually, this is unpublished, but we actually have some really good biological data to show that this approach could work in theory. You know, so we've got a few projects looking at this side of things. You can also just make these, which is nice, in E. coli, express them, purify them, and test them. So it's almost like another antibody alternative, which is nice. This is really cool. And then obviously the diagnostic approach to parasitic disease, which I will focus on in a sec. We focus mostly on making the proteins and studying them. And because obviously extracting them from a Cat3 organism to then do biophysics on is not really feasible, we tend to go down the cloning route and express them in different systems, mainly E. coli, where I grow them up in bioreactors and then purify them out. Here at Babs, I also do mammalian culture too, where things are complicated, difficult to fold, express, etc., etc. Or, you know, if you're making like more the antibody side of things, we go mammalian cell culture for that. And also, you know, the purification pipelines. We can also use the crystallography suite here for setting up, you know, automation with the mosquito and set up crystals and trays and things like that. We obviously can't image them here, but we collaborate. We have like bag access down at Diamond with DLS, so we can go down to Diamond, shoot those crystals, you know, do a lot of the structural biology work down there. Yeah, you know, so some of the collaborators, Leicester Uni, Oxford, Diamond, Nottingham Trent. So, you know, we have some of the research complex too, while it's, you know, things are going. And some of the techniques that we are covering in the group are like some of the atomic and high resolution type things. So crystallography, cryo-EM, NMR, like SACS, HDX max spec over in Oxford. So we do a few of these, you know, we handle some of these techniques with collaborators. And then we also have a bit of expertise in the biophysical characterization of the, I guess, the less high resolution side of things, but more for properties, dynamics, stability studies, things like that. So the list continues, but these were just some notable things that we've done some work on so far. So the actual project that my contract was on was focusing on using one protein, TC24, which is a flagellar parasite protein, which has a existing partial crystal structure. And this was a vaccine candidate, which was shown to elicit nice response in animal models and, you know, protects the parasite. But it was kind of wholly more or less ignored because it was tricky to work with, doesn't express well, it's a bit unstable, you know, and people kind of left it alone, and it wasn't really handled much. But then we decided to take an approach where we computationally designed variants that should have much better expression and stability properties in silico. And we decided to try and use that to use this protein as a diagnostic tool, particularly because it's flagellar protein, it's in the blood stage, it should be able to be picked up at a blood stage with something like a lateral flow test, as is seen here. It's also super conserved across loads of different strains of this parasite. So it's a nice, really nice model protein to work for this diagnostic approach. So yeah, we designed panel of variants, they shortlisted these, and I've only shown 3, but there was more. We just ended up doing a bunch of techniques on the best ones, the ones that we could express the best, and that had good in silico properties. And these were variants, named variants 1 to 3. As you can see here, the wild type doesn't express very well, but when we express these computational variants, they actually up the expression levels quite a lot, which is really nice, because this shows these were all done under the same conditions, so we achieved much better expression. And then we scaled this up and made them in bioreactors, so that we can do all the biophysical stuff, and we can also like cheaply make a ton of this protein, because lateral flow tests require very, very small amounts of antigen once you've optimized them on the strip. We wanted to see how these were basically. So we did some segmals down over at the research complex in Diamond, and yeah, just had some basic properties stamped, like the hydrodynamic radiuses, molecular weights. They were very monodispersed when expressed, which is really, really nice to know. And we worked out some of these basic characteristics. Then when I subjected these variants, I wanted to see how stable they were. Can we claim the in silico stability properties in reality? Does that translate? So I did a bunch of tests to see how they would behave. I guess the real gold star here was we did some 1D NMR in solution, where we basically set them up at a set temperature and just measured their 1D NMR spectra over a period of time. And it kind of works like for those not familiar with it, it works a bit like a fingerprinting exercise. So you got day 0 and day 70, right? And the wild types, their fingerprint, the peaks and the proton groups change quite a lot, right? As you can see here, so that shifts. But then in the variants, their stability seems to be quite nicely retained in comparison to this. I mean, this is fairly qualitative, but it shows as a proof of concept that they are more stable. It showed some real nice results, we think. So these are variants 1, 2, 3 versus the wild types without their molecular tags and just with the molecular tags. So just to make sure that the tags aren't contributing to their stability, it has to be from the amino acid modifications that were made. We then wanted to look at their melting temperatures to see if they've got thermodynamic stability. We looked at that through nano-DSF, differential scaling fluorimetry, or thermophoresis, depending on how you look at it. And as you can see here, with the variants and the modifications added versus the wild types, which are here, you start to add variations and mutations, and it increases their stability. And these are all in the same buffer, etc., etc. So those stability properties are thermodynamic. This was also looked at via CD, and it correlates really nicely. So both techniques agree with an improvement of stability. I also ran them through a bunch of freeze-thaw cycles. So I freeze-thaw cycled all of the variants and their wild types 5 times. And we used something called feeder bio, which is a flow-induced dispersion analysis. So what we're looking at here is changes in hydrodynamic radius of the monomer in solution. And what's quite nice is the freeze-thaw cycles didn't actually have a significant difference when we analyzed this statistically between freeze-thaw cycle 0 and 5 and in between. So the molecules essentially stayed within the hydrodynamic radius range. So, they were fairly stable with freeze-thawing, which is really nice. We also, this needs expanding on, but we also lyophilized or freeze-thawed them with the thought of, if we can freeze-thaw these, you can reduce cold chain dependency, so make this much more affordable for obviously countries that can't afford this stuff that much, and you can ship it around the world to hot places like South America where it's going to be used. And the fingerprinting, non-lyophilized and lyophilized, I mean, it matches quite nicely once these were resuspended and run under 1D NMR. So it looks for now like they are the same. Do you see what I mean? Like this is preliminary. This is very kind of needs expanding on, but I mean, the traces look very similar. It suggests that they are retaining most of their structure in the solution, which is really nice. Well, I'm very excited about that. Of course, this will be purely academic unless you check it that it actually works. So at least 12 historic patient samples were tested against the variants and a wild type to check that they retained their antigenicity, at least via ELISA. So these were CHAGAS patient samples, human sera, and yeah, I mean, these are all they're different. So they're different strains in southern hemisphere and northern hemisphere, but compared to the wild type, they still retained antigenicity in addition to all of these stability improvements. This we're currently expanding on with, like, we've got a collaboration in Barcelona where we're looking at getting 100 patient samples, all tested under different conditions, so that we can, you know, have a really nice big cohort of patients, and also not historic, but, you know, in the clinic patients, and see if they test positively when checked against the variants. We also collaborated with a company called Chorus. This was done through 2017, which is a charity set up by, you know, a professor in Kent, Barry, really. And we put, well, this was proof of concept, but we put the molecule onto a test strip to see if it would test positive. And it did test positive, particularly with Southern Hemisphere strains. As you can see, the test strip should have 2, and this had 2. And there was minimal to no false positive detection from patients with other parasite diseases like malaria or leishmaniasis. So this way it works. It works very nicely. So now we're just looking at basically rolling this up, expanding it, making the prototype, the lateral flow test, work essentially with a much larger group of people, so we can start to roll that out, hopefully. And yeah, these are just some of the members of the group, PhD students, Alicia, you already know, and some of our collaborators that were involved in this process, especially the people down at the London School of Hygiene and Tropical Medicine who did all the ELISA work and collaborated with us, and Barry, who started 2017 charity. Yeah, so that's it. I hope I didn't blow through that too quickly. Yeah, thank you. There we go. Okay. Oh, you're muted, Steve. Just... Steve, I've got you on mute.
You didn't hear the applause. There we go.
Yeah.
I was muted, yeah. Thanks, Chris. And I said good to see Giuliano, the CD guy down at at Harwell.
Yes, it was great.
Yeah, and you know, we had a similar sort of experience with the glyco vaccines being developed against meningitis and things. We did with the vaccine people at GSK and Rixensar. But what I mean, it looks all beautiful, and these TC24 vaccine-based proteins being produced,
yeah,
they're all E. coli, yeah.
Yes, yeah.
So and there's no glycosylation problems in the native. They're not glycosylated.
No, funnily enough, I've made them in mammalian cells, because with the thoughts of, you know, if this is going to be used as a vaccine candidate, you know, I mean, we can't make it in E. coli, you know, it's not going to fly. So our choices are then obviously, you know, yeast, bacilla, or mammalian. But the interesting thing you mentioned that is I made them in mammalian, then did an ELISA, and I did get a drop in detection from mammalian. And I suspect it's got that glycosylation problem. You know, something's hiding the epitope. You know, the interaction with the antibody that we had raised specifically for this protein from E. coli. So these are definitely things that we can start to look at, I think, if it goes down the vaccine route, particularly. Because diagnostic is, the way I'm thinking of it is, it's not necessarily a problem because it's not going into anybody. So as long as it can pick up antibodies,
yeah,
not an issue. You know, that's minimal development there. But as soon as it becomes therapeutic, yeah, we're going to went into a whole lot of questions.
Okay. Any other questions for Chris? Rich?
No, I joined halfway, so I missed the beginning. But otherwise, I did see.
We recorded it anyway.
Amazing
work. Like yours, we recorded it, Rich. So I'll get that out to the team. Gordon?
No, I'm okay. Didn't have any questions to ask. I was just quite interested. I'm interested in lateral flow tests. So I teach about lateral flow tests quite a bit. So
the COVID thing all came out, the C&T, that was interesting, that these test strips used for
I think it's
all the buggies, basically.
yeah. It's I want to obviously I want to thank COVID for that, because I think it's become very easy to mass produce these now, which is nice, because obviously if you can bring a test down to cheap, the issue is cheap. We need to make this cheap for rural communities in South America, which is where it's predominant. So I mean, you don't want somebody spending 20, say a test is $20,
and also there's going to survive the process, the storage, the temperature, all this sort of stuff that happens between manufacture and delivery to people on the farm in the middle of as you said, South America.
That's what we hope to. I've shipped off at least the preferred variant, which is the middle one. FYI, I mean, this is insider knowledge, but the third variant, even though it's got the highest stability, it has some aggregation problems. So your yield immediately drops unless you do loads of optimization. So there's different routes you can take. But at least variant number 2, we've shipped off to the collaborators to see if that picks up serum antibodies. So that would be awesome. If you can just shipped it freeze dried, you can just resuspend it and it works even better.
Check aggregation, from your MSc course. What the gold standard method is. That's the
that's a good point.
Yeah. Anyway, we've got to finish very shortly. Anybody else got a question for Chris?
Yeah, Richard's got his hand up.
I've got a quick one. So it's regarding the aggregation and all the tests you did. I'm assuming you did them in a vial, in sort of PBS or some sort of buffered solution. Is there a difference between any kind of stability issues you see in that situation, and if they are in situ on a lateral flow test?
That's a good question. With the formulation and stability side of things, we did a very small panel of buffers by a CD. The preferred variant, actually, if you've got 1 second, I can literally show you. I've got the slide, but we haven't seen much of I might have this just handy. We didn't. Oh well, okay. Of course, I haven't got it handy.
So when it's on the lateral flow, is it completely dried out and just ready to go when a fluid then comes on and rehydrates?
Yeah, basically. Yeah, that was the way the prototype was tested. Yeah. I think that's. But good you mention that, because that will be part of one of the grants we're putting in, is to do all of the development work around. Basically the tests. I mean, if it can be dry, great. That would be, for me, that would be ideal, I think. Keep it happy. Now it's in my head, back to the formulation side of things. The nice thing about these is in the CD side of things, we really didn't see much change between at least common buffers. So I tested a bunch of common buffers with Juliana, and they kept all the secondary elements and tertiary. There was very small shifts, but funnily enough, things like PBS, I think PBS was one of the best buffers, which is almost like.
There's nothing wrong with it. People.
No, I'm chuffed, I'm yes, because we've done most things we've done in PBS, so minimal tweaking, which is nice and cheap. So it's just cheap, But this is I mean, I'll keep everyone in the loop too. I think if we hear positive results come out from the grant applications for the development.
And Richard, an ideal system of multi-sig too to be used.
Still need to solve the double molecular weight problem, but we're working on that.
Try looking with a factor of 2, Richard.
Yeah, I know. I'm whipping the coder to look into it.
Yeah, I can look at it for you if you like, no charge. Right.
Do you know what? Just before we close, I would say I'll definitely talk to you guys because we're waiting to hear from a big grant. big development grant. Fingers crossed that comes through. But if that comes through, there should be some scope to make some things in mammalian systems. Some of the stuff we will use will be from mammalian, and then I'll definitely be looking at glycosylation things. I mean, there's changes happening to the molecules. As soon as I switch system, start behaving differently, to be expected. But
if there's some short change from the big grant, then I recommend investing in a SecMel's photometer.
Yeah. Well, it'd be nice to have it here, because that would be the next. After I've purified it, I would like to have everything through SecMel's, because then at least you have a few, answers quickly ticked off. And
as Gordon intimated before, we do need a photometer, absolutely. If you've got to pay 50 grand for a new photometer, you have to get it from a grant. It's as simple as that. And everything else is working fine here. Okay, great. So the next meeting will be in a fortnight's time. We haven't sorted a speaker out for them, but if everyone's happy, we'll keep it at 4 o'clock, which I think is okay. Brilliant. I'll get this recording out. Okay, good stuff.
Brilliant. Thank you very much.
Thanks, everyone.
Thank you. See you later.
Bye.
Okay, bye.
Bye.
Bye.